Welcome to Town Hall. Welcome to summer. One of the things that I've enjoyed over the last, I don't know, week or so is like watching all of the trees come out, the leaves come out. All of a sudden, everything is in bloom. A couple of weeks ago, I think I showed off some radishes that was growing here in the office. They've actually already bolted. I've got one that's flowering. So, I can see a little courtyard with a couple of Japanese maples and things like that. Anybody got anything flowering or interesting happening out their window or in their garden? Looks like Bob's got a roof.
I have a large garden. I have radishes in the ground, I have beets in the ground, spinach.
All right. All right. Well, those are good eating early on, right? I mean, like there's nothing like I don't know. Like my beets never made it to beets. Uh, we always just ended up eating the greens, in part because in clay soil, which I guess this place has clay soil, right?
I'm not sure in general. I'm
No.
north of here and fairly closest to the lake, so we actually have really good soil where I am.
Oh, okay.
I've been really impressed.
All right. Well, back in the garden that I had, it was all clay, and even after years of working on it, I would get this misshapen little beet that looked like, you never know what it was actually going to look like. In fact, it wasn't that different than that. There are people who grow gourds in boxes to have them conform to the shape of a box, right? You can put faces on a gourd and things like that. And more or less, that's what would happen to my beets. It would look like some piece of clay or rock had misshapen my beet. Um, so we just ate the greens.
My radishes were pretty sad last year because I won't thin them because it's emotionally difficult. So they were all like flat on one side and
All right. Well, that's enough of the small talk probably for now. You guys don't need to hear me talking about gardening any more than you absolutely need to. And I think we've reached that perhaps, yeah, the pharmacological levels of that. So I want to welcome our panel with me. We have Valerie Chai, Director of the Center for Genetic Diseases, Marc Glucksman, Professor and Chair of Biochemistry and Molecular Biology, Erin Howe, Assistant Professor of Biochem and Molecular Biology, Christopher Kywe, uh, who is a PhD student in microbiology, immunology, and Bob Marr, assistant dean for research in CMS. And we're here to talk about National DNA Day, which is, I think, tomorrow. But like it's DNA Day every day, right? I mean, but we do take some time in particular, given our namesake, and to think a little bit about DNA, how we know about it, what we know about it, and how people are working with it. So, but before that, let me do a little bit of update just on what I've been working on or what's been going on here for the last week or two. No town hall last week because of the lived experiences event from DCCO. It was great to hear people's stories. So thanks to everybody who went to that and who helped make it happen. Last week I was in Springfield to meet with some of our legislators and also to testify about cash for moms and babies. So, which was a fascinating event. On the way to that, because they were about 3 or 4 hours late, actually 5 hours late. Was supposed to testify a little after 5 in the evening, and I testified a little after 10 at night. But along the way, I got to listen to their discussion of the budget for Medicaid and things like that. I did get to watch a little bit of the sausage being made. It was fascinating. And they're open to the public, so if you ever get a shot and you're down there, it's an interesting experience. But it was great to represent RFU, talking about ways that institutions can help moms and babies. It was also last weekend at one of the admitted students days. It was great to be in a room of people coming to join us and along with their folks or their significant others. And I just want to thank all the folks in admissions who make that work and what a great experience they have. Tied to that, the student ambassadors, we had a celebratory lunch for them this week, and they were there in force for Admitted Students Day. It's a room full of really enthusiastic students who love RFU and love helping people think about how they're going to join them in health careers. So lunch was a lot of fun. I think it was yesterday. Um, and just so much enthusiasm, which in the middle of budget season, somebody like me absolutely needs. In terms of the budget, I mean, you guys got I think a email from Emily. We're coming to the end of the year, which means we want as much of your stuff that belongs in this fiscal year to be in this fiscal year. So if we don't get the service or we don't get the product, then it goes in next year. And this is a good year to put your budget stuff in. So, and then related to that, for next year, Gavin has worked with me on a system that the colleges and a few other units will be able to save some money. So we'll talk about that in the future, but basically the ability for folks to create a little bit of a savings account to help invest in things that they feel like they need to invest in. Of course, that also means that when you want something, we're going to ask you, to go in with us on it. But I'm hopeful that this encourages people to save some money, not spend it if they don't need to, and keep it around for investments in faculty, students, and programs. So that's one of the things that I think I early on thought we needed to work on. I really appreciate Gavin's work to figure out how to do that. I also got to go to the M3 clinical posters. So these are the third year medical students who put together a poster, and I think I got about half a dozen tours of posters. I particularly enjoyed the internal medicine one since that's what I do. Somebody had a disseminated histoplasmosis that was a great case. I just didn't really enjoy talking about that. The others were good too, so not to get, play too many favorites. Um, so that's pretty much what we've been working on. We are thinking a lot about the budget. We have had $1 million to close last month. We're making some progress, so I appreciate everybody's diligence around that. I'm pretty confident we'll get to a break-even budget. Well, there's some work for us to do, but that's really the first step on us being able to make more investments and do some fun stuff. So I appreciate everybody's work on that. But this is going to be more fun. This is DNA Day, and I'm gonna kick this over to Marc in a second. But we are, of course, Rosalind Franklin University, the only university in the country, as far as I know, named after a woman scientist. She's up on my wall. And actually, last weekend I met with her niece, also Rosalind Franklin, who is a former board member of ours. It was always interesting to talk to members of that family and think about what that legacy is for them, but also for the rest of the world. And anyway, it's been one of the fun things about this job has been getting to thinking a little bit more about that. I have a few stories about the Human Genome Project and some things like that I know Marc was involved in. And Marc, you've got a brass version of Photo 51 over your right shoulder. So let me just hand it off to you for a little bit.
This picture is this is a statue of Rosalind Franklin with Photo 51 that's right outside our main entrance from a different angle. So anyway, DNA Day, which is officially celebrated tomorrow, is a big event on the 4th floor of the IRP. It's a big event from our center. It's a major holiday. Personally, my scientific lineage and journey has a direct connection with both Rosalind Franklin and the Human Genome Project that sequenced the 3 billion letters of the DNA alphabet. So Rosalind Franklin is my great aunt scientifically. So my advisor's advisor was in Dr. Franklin's lab and a best friend. And when I spoke to Rosalind Franklin, the real niece by genetics, I had to explain that, but it was a lot of fun. As far as the Human Genome Project, I collaborated with the laboratory of John Martinetti back when we were at Mount Sinai School of Medicine in New York. And we published, it was either the first or second paper with sequence data from the Human Genome Project in early 2000. And that was from chromosome 22. And what's the significance of that? That's the smallest chromosome, so it was the easiest and the first one to sequence. And it's the smallest non-sex chromosome. And it explained the cause of a bleeding disorder called May-Hegland anomaly. And coincidentally, Dr. Martinetti is on our campus today, and he's here to celebrate at the white coat ceremony of his daughter, who's enrolled in one of our programs. So coincidences abound. And I'll stop there.
Well, so do you want to just say a little bit about what Photo 51 is? Why is there a Photo 51?
So other than a cool logo, and I noticed that you're not, okay, you got it. Okay. So other than a cool logo, this is an X-ray pattern. It's not a direct photo.
Mm-hmm.
And this was taken with an exposure. So if you think of it as sort of as a camera, it took over 2.5 days, 68 years ago this week, to take this iconic picture. Today at Argonne National Laboratories, where RFUMS has a long-standing presence and we're a scientific partner, which is 20 minutes south of the airport, it would take 1 or 2 seconds to collect this type of data. So essentially what this did was, with geometry and chemistry constraints, this shows the double helix structure of DNA. We teach this to our medical students and BMS students, and that's actually Dr. Carl Carell who does that. And what's so special about X-rays? We talk about X-ray diffraction. Essentially, it's a good way of depicting regularly repeating structures that create spots or lines on a detector. So this pattern tells us a lot about DNA, and coupled with model building, it's the double helix. And to show how important this is, this is a copy of last summer's, and this is a very true and depiction of it. So it's like a spiral staircase. And if you take a look outside one of our buildings in our courtyard with all flowers, you will see a 30-foot, it's at least 30 feet high.
It's like 3 stories, yeah.
Yeah,
4 stories.
exactly. So each of these, the details gives you the geometry of the of the helix. It says it's a helix, but it's a double helix. So it's 2 winding staircases, right-hand turn.
So check me on this, Marc, but fundamentally, the way you do X-ray crystallography is you make a crystal, which is a repeating version, right, of something. Water, if it's ice, or various minerals that form a quartz crystal and things like that. And then when you shine X-rays at it, which are a light, you get a pattern out the other side based on the stuff that's inside and where the atoms are. And the reason that you use X-rays is that they're very short wavelengths. And so you actually get essentially the diffraction of that light. And then you have to solve the picture tells you something, but they actually go through in the old days with a ruler and things like that and measured those distances and calculated up what that meant for where the atoms must be. It was the only way to get a sense at that time of how the atoms were really arranged. Does that sound about right? You're an X-ray crystallographer
we might recruit you to teach this in our graduate course. Yes, no, that's very accurate. And it turns out that the second civilian use of computers after World War II was because there's so much data here. The only thing that's a little different is this is not X-ray crystallography, because you can't make crystals of DNA. They are very long and fibrous, so it's fiber diffraction.
Gotcha.
And it's all about repeating units. This is not one strand of DNA. It's probably somewhere on the order of 1 billion strands that have been tried in parallel. So yeah, we don't have to go that deep in the weeds. So
yeah, well, we can go into the history of some of that in a bit, because of course it follows on the alpha helix and proteins that have been solved before that. But Valerie, you do DNA work. And so one of the reasons for this panel is to maybe give people a little bit of a sense. Oh, and I want to say this is always more fun when there are questions. By all means, put your questions into the Q&A. Somebody is telling me that Lake County does, in fact, have a lot of clay. That's what I thought I saw on the ground. In any case, Valerie, you use DNA in your work. Can you just talk a little bit about that and how you think about DNA in your work?
Yeah, happy to. So my work directly works on DNA, and Marc mentioned that it's the double helix structure of DNA, and what we study is because this DNA harbors all the genetic materials in your cell, in your body, and how this genetic information is faithfully passed to the daughter cell without getting mutations or alterations or any kind of things that can lead to diseases. So after the double helix DNA structure was discovered, actually people started to think about how this DNA is passed into the daughter cells. So that comes to the DNA replication concept. So during the DNA replication, this double helix, which has 2 strands of DNA, needs to be copied by the machineries that involve the proteins, enzymes in the cells. And this copying needs to be finished at a very limited time during this cell cycle. We call this cell cycle. And after that, the 2 strands are duplicated, and then the duplicated DNA can be separated into the 2 daughter cells. However, this replication stress is not always smooth. Just think about it. So the DNA strand is quite long. So all the DNA inside our cell is packaged into this tiny little space. However, if you stretch it and to make it linear, it can equal to like 2 meters long per cell. And if you count how many DNAs we have in the whole body and make a rope out of all the DNA, it lasts probably from the Earth to Moon and then come back. That's a lot. How many times? So this super long DNA. Okay, so when the cells are replicating the DNA, you can imagine that this DNA process is like a zipper. So you open the zipper, and then the proteins and the enzyme come to each strand of the zipper and try to copy, right? However, as I said, this is always not a smooth process. And sometimes it's due to your the environmental exposures. Sometimes it's due to your regular cell metabolism that can cause the zipper to stuck. And when that's stuck, the cells need to resolve this. Otherwise, the DNA cannot be duplicated. If it's not resolved well, and then the DNA actually can break, that can lead to all the downstream events I'm not going to talk about, too complicated. And then, there could be mutations and the broken DNA leading to chromosome fusions and all bad things. So in fact, we now know that genes involved or the proteins involved in DNA replication are very important for preventing the cancer and also for preventing the diseases like neurodegenerative diseases and developmental defect and even the premature aging. So what we do is to use the molecular biology methods and biochemistry as well. Sometimes we collaborate with the structure biologist to understand how cells actually can keep the DNA replication process faithful, and that the genetic materials can be faithfully passed into the daughter cells. At the same time, when the zipper stuck, we also have a term for that called replication stress. It turns out that a lot of cancer cells, you can target this process and target that replication stress to make the cancer cells more sensitive to current treatment. And we call this targeted therapy. So we are, by understanding the mechanism of how the replication stress is resolved, how the DNA replication is maintained, we can either help to prevent the diseases from being developed or to provide some insight on designing more effective treatment.
Yeah, so Michael Rosen has a question sort of about us differentiating some of the things that are going on here. So, but in the replication of cells, right, it's really DNA separating, new DNA forming on each strand. And you
Yeah.
can just imagine that zipper piece, right? And there
Right.
are not only are there special proteins that help make sure that the right base joins up. There are
Mm-hmm.
there are proofreading systems in there to make sure that there's not a mistake that gets made and to go back and fix it and it compares across the two and then you go these things are pulled apart and you
Yeah.
get like there's a whole fancy system around this replication which is sort of unregulated in cancer cells, right?
Correct.
and
Yes.
and then there's the central dogma which comes from
Mm-hmm.
Francis Crick in the
Mm-hmm.
late 50s, right, which is you have DNA, then it
I
opens and RNA forms off of that. And RNA gets turned is then what your body uses to build proteins off of those
correct.
little groups
Yeah.
of three nucleotides, and then your body helps
Mm-hmm.
metabolize the protein from there. And
Mm-hmm.
that dogma is sort of the central features of a whole range of things that we do, everything from how particular antibiotics work in stopping some of that in particular bacteria to cancer treatments that stop some of that to now we're figuring out that RNA on its own does a range of things.
Right. Mm-hmm.
So it is part and parcel of this whole system is part and parcel of just about it's taken over biology in a lot of ways, which is
Yeah.
the whole field of molecular biology, not to put too fine a
Yes.
point on it, right? Otherwise
Mhm.
it was, and I am a bugs and twigs biology guy, but there was a lot of bugs and twigs biology happening before, and now I once had a bat get into my house during a poker game, and the molecular biologist is like, Doug, you're a biologist. Help us out. And he's like, I am a molecular biologist. He wanted nothing to do with a bat. So the forester in the room had to help with the bat.
Yeah, we do molecular biology on all the molecules, DNA, RNA, and proteins.
Yeah.
Yes, so
Yep.
that's what we call ourselves, molecular biologists. Yes.
Chris, you are a grad student, so you are coming into all of this kind of work. I well, probably almost certainly did it as an undergrad, but can you talk a little bit about what you do and how DNA figures into what you're working on?
Right, Dr. Sousa, I'm happy to share my lived experience as a graduate student. So, my entire project revolves around a double-stranded DNA virus. So, rather than focusing on cells, I'm kind of working more on the pathogen side. But the virus I work with is Kaposi sarcoma associated herpes virus, KSHV, also known as human herpes virus 8. And I'll share a little bit of the history of this virus since I think it's pretty interesting. But long story short, we wouldn't be able to even understand that this virus was present without our current understanding of DNA. So this virus came into prominence around 30 years ago during the AIDS and HIV epidemic during the late 80s and early 90s. So 1 in 3 HIV and AIDS patients would develop Kaposi sarcoma, which was essentially just a pattern of like reddish brown lesions on the surface of the patient's skin. And they're highly proliferative, angiogenic, kind of devastating cancer. Most patients would die within 5 years. And so there was a real sort of arms race to try to understand why do some of these HIV and AIDS positive patients develop these lesions. And so basically what they would do is something called relational DNA analysis, where you can isolate the DNA from like the diseased patients, and then you can also compare that to like healthy controls. You can amplify it with PCR or polymerase chain reaction. You take small amounts of DNA and just make up in really high concentrations, and then you can digest them with these restriction digest enzymes to fragment the DNA. And then depending on how big or small the DNA fragments are, you're able to see them on a gel. Like they show up as like these little bands that are present there. And you can compare the presence or absence of certain bands between diseased patients and healthy people. So what they were able to find is that there were these unique DNA sequences in the Kaposi sarcomas patients that weren't prevalent in like people that just had HIV alone or people that were healthy. So Then they were able to do some sequencing and find out that those sequences within the Kaposi sarcoma patients actually belong to an entirely separate virus, which is what I study right now. And a lot of things that I do focus on trying to understand how this virus causes cancer, specifically how this infection impacts monocytes and inflammation and things like that. But long story short, without our understanding of DNA, I wouldn't even be able to do the work that I'm doing today because we were able to sequence the DNA from these afflicted patients and really just identify a whole slew of pathogenic agents that causes harm in our everyday lives.
So can you talk a little bit about what you do? Like what is it that is you're working with this virus and things like that? What is your work?
So the big problem within the field is that people don't really know how infection with this virus leads to cancer. They just find these sequences of DNA and they don't know how we get from like this infection state to the cancer causing state. So I'm trying to figure out the stuff that's happening in the middle. And one of the things that's believed to drive the cancer development is KSHV associated inflammation, where if you're taking like the blood from these patients, you're able to see elevated levels of like interleukin 6, interleukin 10. Cytokines are families of like inflammatory signaling proteins. And so we're trying to understand how KSHV infection itself influences the release of these inflammatory molecules. And then secondly, we're trying to see if we can inhibit the ability for KSHV to induce inflammation by targeting these water channel proteins known as aquaporins. So if we take these small molecule inhibitors, block them with different kinds of, block the aquaporins, then we're able to see what kind of changes are seen during KSHV associated response. So.
Oh, great. So, I mean, this is, I'm stretching my virology knowledge, but the herpetic viruses and human papilloma virus, those are, double-stranded DNA viruses, which means the virus itself has a double-stranded DNA like we do. There are other viruses that use RNA and things like that, but these double-stranded ones have are notorious for being linked to cancer, right? So whether it's Epstein-Barr and Burkitt's or human papilloma and cervical cancer or the herpetic viruses and KS or one of those other sorts of things. I mean, the viruses, because of their genetic structures, are often related to the diseases we have because they move into our cells and take over our machinery as though their genetic component was a part of ours. And actually, we're running around with a bunch of virus genetics floating around in us, probably.
Right. So a lot of these different viral gene products, they're actually meant to enhance the survival of the infected cells, right? So you talked about papilloma viruses earlier. So they encode proteins that like target retinoblastoma or like different cell cycle arrest proteins and like cell cycle inhibitors in order to sustain the proliferative state that they need in order to replicate their own genome. And as a byproduct of that, that causes cancer. So different types of viruses have various mechanisms
that cell cycle is what Valerie was talking about, right? As the cell moves through different steps in replication, a whole bunch of our cells' lives are not actively replicating, right? But then there's a section where they are, and cancer cells in particular spend a lot of their time in that reproductive part, which is where the damage and the opportunity lies, right? Depending on whose side you're on, as it were.
Yeah, exactly. So viruses have a bunch of different ways to kind of mess with that process, and sometimes it can be more dangerous compared to others.
Right. So, Erin, can you tell us a little bit about your work in DNA?
So my work mostly doesn't focus on DNA. We mostly look at how protein gets trafficked inside of cells. But one way, so we study breast cancer and breast cancer metastasis. And one of the ways that, going again back to, since Valerie's the expert here, what Valerie, one of the elements of DNA and how it influences, one of the things that we work on is the ability of the immune system to recognize cancer cells. And so you probably heard, what I think about a year ago, there was a clinical trial where 100% of rectal cancer patients were cured with immunotherapy. And what turned out to be the case there is that all of those patients had mismatch repair deficient cancers. And so that leads to a lot of what we call tumor associated neoantigens. And so when you think about how your cells all signal to your immune system that they are self, that they are you, they do that by putting little bits of protein of all of the different proteins that they make inside of themselves on the surface of cells. And so because cancer cells have so much genomic instability, their DNA is getting broken. They should be making all of these aberrant proteins. And in fact, they are, but most of the time, that's actually not leading to an immune response. And so, one of the things that we study, and not at the level of the DNA damage, but sort of upstream of that, is how that signal essentially gets lost going from the damage to the DNA to the ability of those neoantigens to be displayed on the surface and recognized by your immune system.
Well, I mean, there is a little bit of a evolutionary arms race, right, between cancer cells and us. I mean, fundamentally, if you were a cancer cell, you could be a cancer cell that was putting all that information on the surface, and our macrophages would come along and clean you right up.
Right.
And so, if that's going on, the ones that survive and then have the opportunity to proliferate are not going to be doing that.
And so there's a lot of balance because if your cells aren't signaling that they are self, then I think it's your natural killer cells are supposed to recognize them
Yep.
and kill them at that point too. So they have to balance between basically showing the immune system that they're broken and just disappearing or trying to disappear entirely.
Yeah, and when I was a wee lad, there was a lot of interest in programmed cell death, right, which was essentially a mechanism by which the cell's death could be triggered because it wasn't working right. And there are mutations that successful cancer cells have that overcome that, which is why there's a whole class of immunologic chemotherapy agents, which are anti-programmed cell death 1, anti-PD-1. There's a Nobel Prize from, I don't remember, 15 years ago or something about those drugs, where the drugs actually then make it possible for immune cells to go in and trigger that system again, which is fascinating to me, right? But it is all basically DNA evolution. The DNA is screwing up, and then you get this phenotype that comes out afterwards, and because it's more or less the same genotype, but the phenotype is different for these cells, and then we can or can't clean them up. So what sort of stuff are you finding?
So I am a new assistant professor and we've only been here 2.5 years, so
Hmm.
we don't know a whole lot yet. Um, but my graduate student, Kathleen, does have some really interesting results that she actually just got last week, sort of looking at, and again, I don't want to get too much in the weeds, but has found a couple of Rab interacting proteins that are really influencing the way that T cells, so cytotoxic T cells,
Mhm.
can recognize the breast cancer cells.
Interesting. Yeah, that's pretty cool.
It's
At some
early
point, we should
days.
actually, I'm going to go to Bob, and actually, then we should actually talk about how people become a scientist. There may be some folks who don't know what the difference between grad students, postdocs, and what an academic lineage is that Marc was talking about. But Bob, I want to move over to you a little bit. You're uh, just DNA in your life, maybe not counting internal turnover.
I mean, I would say that of what I do here, the most fun part of what I'm doing, that I still find the most fun, is the work I do directly with DNA.
Mhm.
So, I do a lot of recombinant DNA technology. So I'm actually holding DNA in tubes a lot. I even in my freezer, I think of it like a toolbox. I have these blue freezer, basically portable freezer units, which is full of restriction endonucleases, ligases, polymerases, phosphatases. Did I say nucleases? Yeah.
Yeah,
Well,
yeah,
not restriction, but even and endonucleases that will chew back things like that. All these things together can be used to, whenever I need to do something, I can rearrange a gene, I put a different promoter on it. I can basically change domains to change the localization, put it on the cell surface, secrete it, and make mutations, and basically, increase expression, decrease expression with RNA interference. So there's, I'm always in awe of just how powerful the technique is, and it's good for research in vitro. Just, what does this do? Does this enzyme do this? So you overexpress it in a dish, and then you have some assay. So that's the most basic application. Then the others are gene therapy. I do a lot of viral vector technology work, so I make lentiviral well, I have my PhD was in adenoviral vectors, but so we make vectors that can be used, that we test to see if they could be therapeutic, like in an Alzheimer's model or something like that. Also, cellular reprogramming. When you express specific transcription factors, you can change the fate of cells. You can use that as a research model, or even to pursue regenerative medicine approaches, say in the central nervous system or something like that, by reprogramming glia into neurons. So yeah, there's a lot of potential, a lot of different things you can do, and I apply it mostly to Alzheimer's disease, and use cellular programming to study, either develop therapeutic approaches or to study Alzheimer's disease.
Okay, so let's talk about this a little bit more. So one of the things when Rosalind Franklin was working with DNA and we were trying to figure out what was in there, they knew that there were these 4 bases that showed up, because when you broke it, you could find these things, and they knew that there was this, ribose. There was a huge amount of ribose forming this, probably this structure that sort of held stuff together. And they were trying to figure out, well, how does this whole mess go together? Because when they would break it out of cells, it just looks like a pile of snot, right? Yeah. And snot has a lot of DNA in it. So, but now it's almost as though it's a series of bricks that you're just putting together sort of in any way that you want. And you have different ways of, it's actually kind of amazing. So one of the questions is sort of like, what is gene therapy? What is CRISPR? Like, and we don't have to get too far into all these things, but, Chris was talking about PCR, and there were some conversations about recombinant DNA. You want to just talk a little bit about some of the major technologies that most of them are, under 30 years old. Some of them are much more recent, but like the ways that we can, work with DNA at some sort of simplistic level for folks.
Right. Well, I mean, first off, gene therapy. Gene therapy traditionally is the idea of, if there is a defective gene, of replacing it with a working gene. That's probably the simplest approach to gene therapy. And there are some of those, there is some of that. Actually, some of it's even getting into the clinic now for like sickle cell and adrenoleukodystrophy. It's actually happening now. And so what I've often used it as a glorified expression system, almost a drug delivery system. So if you need more of this enzyme, you bring it in, you overexpress it, or and you try to get it to cross the blood-brain barrier or something like that. So that's that's basically gene therapy. You mentioned CRISPR. There's a lot of potential in there. A lot of people have heard about CRISPR. So, and even that's in the clinic now. Basically, you take someone's hematopoietic cells, you CRISPR edit them to correct a genetic deficit, expand them and put them back in, and hopefully it repopulates the hematopoietic system. So a lot of potential power there. CRISPR allows you to very selectively, I mean, the concept of what CRISPR does is allows you to make a cut at a specific location. It was around for many years with TALENs as well. They could engineer proteins to do that, but CRISPR just is easier to use, and it's more reproducibly successful. It's a very potent system for a lot of things. And then they've adapted that to CRISPR-A and CRISPR-I, so you can actually use CRISPR to upregulate a gene or downregulate a gene, not to cut the genome, but just to upregulate and downregulate genes. So again, I'm always in awe of all the potential there. This is always so much potential in recombinant DNA technology and things like that. PCR, I mean, it won the Nobel Prize. Actually, in my PhD lab, we had a PCR machine that was signed by Carrie Mullis, who was who won the Nobel Prize for PCR. You know, that I mean, it's in everything. I mean, it's a forensic DNA, you know, analysis. Anytime you need to amplify that, the unique thing about DNA is it can be copied, so you can amplify it from a single molecule. You can get that information. So PCR has been, and for recombinant DNA technology, PCR is widely used. And then, you know, if you want to talk about things that are impactful, like in the clinic, you know, CAR T technology that's based on a lentiviral gene transfer to a person's own autologous T cells, and it's very it's been very successful in the clinic. So I will say one last thing. I've been doing this for a long time, like 30-ish years, and I think it's been moving very slow. I know it's exciting now, things are coming out, but there's been some valleys of death for the gene therapy fields, but it's and there are
literally
complications
some valleys of
sometimes.
death. Like, they took out some people in the first times they were trying it.
Yeah, I mean, there's been, and yeah, well, there's the famous case of some deaths in clinical trials with the adenovirus vector.
Yep.
I remember when I arrived in San Diego to start my postdoc, I turned on the TV and my postdoc supervisor was on TV in front of Congress testifying as an expert about that case, that specific case, the case of the death.
Yeah.
So it was pretty interesting stuff. Oh, and one last comment about lineage. Marc had mentioned a lineage. I did meet Francis Crick when I was a postdoc at the Salk Institute once.
So Marc, you want to talk a little bit about lineage and what that means? I mean, it's a bit of a side piece for us here, but we've got it there may be a group of people who don't really know how scientists get made. People go to, as an undergrad, they usually get a scientific degree, then they become Chris and become a graduate student where they get their doctorate. And then in these sciences, people often go for a postdoc, right? Where they spend a couple of years, hopefully not 8 years, but hopefully a couple of years in another lab, getting another set of skills, usually around what they're really going to do with their career. And then you go on and you become a principal investigator of one sort or another. And my dad, who's a chemist, when he retired, somebody gave him his lineage back to, German chemists of the 1500s, right? I mean, like, people sometimes my dad didn't, but people do take this seriously sometimes. So can you talk a little bit about why that's important, or why you guys noticed that?
So it's a bit of a feudal system, not you have to excuse my New York accent. It's not FUTILE, but FEUDAL.
Could be feudal. It could be
feudal. Yes, but we're not going to go there. In that there is this progression, and essentially a lot of it is training. And it used to be, and a lot of this has to do with sort of generational demographics and, baby boomers, and it used to be that, pretty much the way to go was academia. Mm-hmm. And what you had delineated was sort of the academic path. Yep. But now there's lots of different pathways. Industry, for instance, so especially when biotechnology came about maybe 30, 40 years ago, that was a path. And just going by our graduate students, as well as graduate programs all over the country, you can also do lots of other things. You can essentially go into programmatic you could go into writing, scientific writing for companies, for grant programs. You can go to national, you can become a scientist. And we've been hearing a lot about that in a bad way in terms of the government. Associated with HHS and research and things like that. So there are different paths. And then there is what we call tenure, and that's one of the unique things here. And we don't have to go into a lot of, that's a little bit contentious. But when you reach tenure, it's not like you put on your pajamas and slippers and say, okay, I'm good. But it does give some stability and some security. And there's also the progression from the laboratory. Part of what we do is work in a laboratory as a scientist, also teaching in all of our 5 colleges, in terms of our health, in terms of our health related students.
My experience is people will talk about, oh, so and so, even late into their career, people will talk about, oh, they were in so and so's lab, right? There's a whole piece of that where, and there will be reunions of labs, and sometimes when somebody's retiring or ending their career in one way or another, they'll a whole bunch of their grad students and postdocs will get together at a meeting, and there'll be a symposium about the work that they did and that their scientific progeny did. It's a whole subculture in science is how people are related through these various training mechanisms.
I was just going to add, since we did bring up, we are a health sciences university with 5 different colleges. So a lot of what we've been talking about is sort of the hardcore science in the lab. But what I find sort of amazing, and what I find probably the greatest medical significance today, is that we each have unique, 99% of it's the same, but are 3 billion letters of DNA. Now, 25 years ago, going back to the Human Genome Project, it cost $100 million to sequence one genome. Okay. And we all know about technology and dropping a price and computers and iPhones and things like that. So 25 years ago, $100 million to sequence a genome. Today, it's about $100. Okay. So it's 1,000,000-fold cheaper over 25 years to do that. And I think because whether it's technology, competition, economy of scale, this is basically democratization and access to science and medicine.
It's been fascinating. Anonymous points out we have 6 colleges in the question.
Sorry.
Yep, that's all right. It's all right. So, one of the things that's clinically happening is that particularly in children's hospitals, when there's a kid who's not doing well, especially a youngin, they'll do a sequence and they're just looking, right? It used to be when I was a pediatric clinical student, every neonatal intensive care unit, neonatal unit had a book of faces. They had a book of drawings of faces of babies because many of the genetic conditions have either down the middle, there's some sort of asymmetry that happens or something weird that happens because of in development. And they would be books and you would go through these pages trying to match up what was abnormal in your baby's, your patient's face with what maybe some of these syndromes were. And now they send off a DNA, they come back with a sequence, and it's not all 3 billion base pairs, but it's a large number of them, particularly the ones where they've identified things. And there will actually be, it's not uncommon for people to find genetic abnormalities, or they're really markers, right? Rather than the actual sequence, but they'll find markers for things that nobody has ever known about before in this kid. When in my prior life, we had a kid like that in the children's hospital. The geneticist happened to have gone to a grand rounds by one of our biochemists who worked on this particular sequence of enzymes. They found there was this kid with a very particular group of problems. The geneticist was Bupp, the biochemist is Bachman, and the syndrome is Bachman-Bupp. And as it happened, they had a small molecule that would downregulate the enzyme, and they had a treatment. It was fascinating to watch, but it was really very much about this relationship between DNA and what we see. Hugo has a question. You can reprogram a cell into a neuron, and people have taken stem cells, fibroblast stem cells, and turned them into neurons and things like that. But can you reprogram it into a more specialized neuron? I mean, can you reprogram a cell into a dopaminergic neuron, for example? I think that we're thinking about Parkinson's or something here. But so how far I don't know if we have anybody here who's
I can
going.
comment.
Yeah, all right, go for it, Bob.
Well, I mean, the short answer to that is yes, you can. And you can there are ways to specify subspecialties within neurons, and a lot of people are trying to do that. And dopamine is a big one for Parkinson's therapy. And it's and I think the question mentioned direct reprogramming, going from a fibroblast into a neuron of a certain type, whereas a lot of reprogramming is going into that induced pluripotent stem cell state first, and then it's easier to get it into the other states. Yep. And that field is it's a very confusing field because everyone presents different techniques for doing that, different transcription factors, different growth factors, and when you have a whole slew of starting cells going to a whole slew of different ending cells, it becomes a very large morass in the literature. Getting to what is really going to work consistently for you with your starting cell type to that dopaminergic, cholinergic, glutamatergic, GABAergic neuron is a bit confusing, but it is being done.
So I want to ask you each sort of what you think is next or what you're excited about in the world of DNA. So I'll give you a moment or two to think about that, because over the weekend I happened to see a talk by Demis Hassabis, who is the founder and CEO of DeepMind, which is the AI company that Google bought. You might know it from AlphaGo if you're nerdy enough. It was the first computer program to beat human champions in the game Go, which has more degrees of freedom than chess, so a more complicated beast for it to handle. And then they went and created a program called AlphaFold, which goes through and takes the sequence of amino acids in a protein and calculates up what the structure would be, because it has been devilishly difficult to figure out what a protein structure really was and how it would change and what you could do to make it change. And so they've essentially jumped into a portion of that central dogma of biology where you get to proteins, and now you could go backwards. You have a protein that you want to have this shape. It can figure out what that amino acid sequence would be. And if you know the amino acid sequence, then you know what DNA, RNA, and DNA sequence you need to have. And you can put that into E. coli and make up vats of it if you want to. So it's a remarkable, and they're taking on different problems in biology, and he's highly aspirational, and I'm not willing to bet that they'll get all the things that they imagine, but the technological calculation of a lot of these kinds of things is fascinating. It'd be fascinating to see where just higher order machine learning, because it's not, this is not ChatGPT, AI. This is machine learning AI. Where that takes us, which is like one of the things that I think will be fascinating. So each of you, we're near the top of the hour, so just a minute or so on what you're interested in seeing happen next in the world of DNA as we approach DNA Day tomorrow. Marc, we'll start with you just because we started with you last time.
Thanks a lot. So in our center, which is the Center for Proteomics and Molecular Therapeutics, there's no DNA in there, except we sort of see DNA as, essentially it's something to make RNA, and we're interested in
It's a part of the machinery. Yeah, it's a part of the machinery.
We're interested in proteins. And even though there's only 20,000 genes, right? There's 3 billion, 20,000 genes, it makes over 1/3 different proteins. And this is because it's not just one protein, but you just can add a whole lot of things to make it completely different. So what we do is we look at, first we look at the DNA, then we look at proteins, but it's about biomarkers. Essentially, it's about diagnosis, prognosis, and potential treatments. And we work with different schools here. For instance, we work with CLEAR, the Scholl research arm, and we look at patients. We don't work with animals. We work with patient material. So this informs us of essentially looking for better diagnostics, and with diagnostics, treatment. That's where the molecular therapeutics comes in.
Great. Valerie.
Yeah, so I think that, so the technology has advanced so quickly, and so it's just amazing. Even, in my 30-something years of research, from initially internet to now, AI, and it initially the traditional DNA isolation and the plasmid and PCR, now this next generation sequencing, and just, so it's amazing, and also CRISPR. So I think the technology is driving the DNA research field. It's pretty much all kinds of research field in a unbelievable way, because it's very hard to actually it's very hard for me to imagine what the technology advancement can do. So just for example, now ChatGPT introduced GPT Rosalind for life science research.
Yep.
And so that dramatically accelerates the drug discovery. And so I personally know the researcher who is using AI to screen for the compounds that cannot be done with traditional methods. And so I do think, I'm excited about all different kinds of the advanced technologies, and also, CRISPR, and we know the CRISPR has advanced a lot, but each year I think there will be some new findings about new CRISPR, new Cas enzyme, that new type of enzyme, and how you can modify the DNA in different ways. I think that's also very exciting because, CRISPR, as Bob mentioned, is easy to do, and it's very efficient.
Yep.
And you can directly modify the cells. So another thing I think at the basic research side is that with all the advancement of new technologies, and the dogma that we have believed for so long, we can find new findings that challenge the dogmas. So just for example, so you mentioned the DNA to get into RNA, and the RNA get into the protein. That's what we believe, right? We also know that RNA can be reverse transcribed to DNA, converted to DNA. But there is a recent study showing that you can use protein to encode DNA.
Oh, interesting.
That's amazing. So it's all those just, researchers.
So complicated.
Yeah.
So wonderful.
Constantly challenge the dogma.
Yep. All right, Chris. Yeah, wonderful. Chris?
Yeah, so actually, Dr. Chai mentioned something that I was going to talk about, how protein can be used as a template for synthesizing DNA, but that's actually like an antiviral defense system. So they found this protein in bacteria that can use amino acids as a template to synthesize single-stranded DNA in order to replace viral DNA or like bacteriophage DNA in infected bacteria. So there might be a way to like kind of maybe use that technology or find a way to express it in human cells, but that could be a possible therapeutic to get rid of viral DNA sequences if we're able to crystallize and amplify that protein.
Great. Erin?
I'll actually let Bob go because we're almost out of time.
Bob, take us home.
Yeah, well, I think, I look forward to seeing more and more FDA-approved gene therapies in the clinic. I think that's going to be exciting. And also related to CRISPR technology, I'm wondering if we're ever going to see a day where people start using gene drives in the environment. They're talking about it. It's potentially, there's ethical questions about doing that with CRISPR, but maybe eradicate certain species of mosquitoes using gene drives or things like that. Whether that'll ever come to be.
All right. Well, I want to thank you all for being with us. Thanks to Valerie Chai, Marc Glucksman, Erin Howe, Christopher Kywe, and Bob Marr. Thank you guys so much for the work that you're doing and for being on the panel today. And thanks to all of you for joining us. Thanks for all you do to support our faculty, our staff, and our students. It is the work of people like you that make the university such a special place to be. It is Friday. I hope you get to have a weekend. Take care, everybody. Bye-bye.